What are endophenotypes in schizophrenia genetics? What exactly are endophenotypes in the context of schizophrenia genetics? This is a fascinating area of study that bridges the gap between our genes and the symptoms of schizophrenia. Endophenotypes are measurable traits that can be biological or behavioral. They sit between the genetic makeup and the visible symptoms of schizophrenia. These traits are not the symptoms you see on the surface. Instead, they are subtle markers that can be objectively measured. They help us understand how genetic risks translate into the clinical signs of schizophrenia. One key feature of endophenotypes is that they are more common in people with schizophrenia and their relatives than in the general population. This suggests a strong genetic link. Another important aspect is that these traits are heritable. This means that genetic factors play a role in these traits. They can also be present even when a person is not experiencing active symptoms of schizophrenia. This state independence makes them particularly interesting for researchers. Endophenotypes also tend to co-occur in families. This means that family members who carry genetic risks often show these traits, even if they do not have full-blown schizophrenia. This can help identify individuals at risk for developing the disorder. Examples of endophenotypes include deficits in working memory, sensory motor gating abnormalities, and eye-tracking dysfunction. For instance, working memory impairment is a well-documented cognitive deficit linked to schizophrenia. It has connections to specific genetic regions, such as a gene on chromosome 1. Another example is the catechol-methyltransferase gene on chromosome 22. This gene affects dopamine metabolism and has been associated with working memory performance and schizophrenia risk. The study of endophenotypes is important because schizophrenia is influenced by many genetic mutations, each contributing a small effect. By focusing on these intermediate traits, researchers can better connect specific genes to the disorder. This approach can lead to a clearer understanding of how genetic changes affect brain function. Identifying endophenotypes can also improve genetic studies. By concentrating on stable and measurable traits, researchers can gain a better understanding of the biological pathways involved in schizophrenia. This could lead to earlier diagnosis and targeted interventions, especially for those at genetic risk who do not yet show symptoms. In summary, endophenotypes serve as a bridge between genetic risk and the complex clinical picture of schizophrenia. They help clarify the biological basis of this mental illness and may guide future research and treatment strategies.